Department of Endocrinology, University of Madras, Chennai. And the co-chair is Dr. Lakshmi S. Pinandar, Professor and Deputy Coordinator, UGC SAP DSA 1, Department of Zoology, Karnataka University, Dharwad. We'll now begin the session, and the chairs will take over from here. First speaker is Dr. Catecholamines, Principal Scientist, Associate Professor, Central Drug Research Institute, his academic qualifications MSc, PhD, Devi Anila University, Indore. His area of research includes female reproduction, female reproductive biology, ovulation, and endometrial receptivity, metabolic and endocrine radiologist by he has received GP Kalwar Award, International Best Assistant Award by Exercise USA 2014. May I request the first speaker to start?
Yeah, very good afternoon, and first of all, thanks, Chair and Co-Chair Dr. Balsubramanian and Dr. Nandar for kind words. And I have got two jobs that I have to keep myself awake and also the audience awake for the post-lunch session. And I will also extend my gratitude to the organizers, SRBC and the Dr. Manli, both Dr. Manli Lalori and Dr. Manli, has to almost putting hard efforts to invite me to come over here, and not taking much time since we have given a little bit quota of time. The thing is, Dr. Mali has already spoken about much of the things which probably will be enlightening to you, and I will try to concise, not to go in that aspect. And Dr. Mali has spoken about some aspects of embryo plantation, their consequences, and I will be touching upon more on the fact that not only plantation, there are issues with pregnancy failure, particularly in our settings. I just talk about the global or the situation over here. So we have worked upon some aspects of epigenetics, where we looked some of the small non-coding RNAs, particularly the mRNA, and we are looking at some more aspects of these microRNAs. And I will be sharing some of these signaling, which we found very fascinating to share with all of you. And just to give some brief information, not only population, because we have issue of population, but at the same time we also have issue of pregnancy failure, and that is really accountable to large chunk of population, because 2.5, and it is defined that 24 week there's a pregnancy failure that we categorize as a pregnancy failure loss. And the ASRM and the societies, like say Reproduction, they have different definitions, but they more or less stand for 20 weeks clear of the pregnancy. And this can be by factors of the uterus, as well as maybe something else which drives the failure of pregnancy. And the pregnancy failure can be linked with, as emphasized Dr. Mali, that the current pregnancy failure can be with endometrial. That means it is probably driving through the endometrial susceptibility that leads to the derailment of interactions with the embryo. Anyway, we have some technical problem. So the current pregnancy failure, having the implantation failure, it accounts not 5% worldwide. Right over there or here?
Yeah.
Yeah, okay. embryo plantation continued largely because the IVF attempt is to make the morphologically as well as genetically good embryo, which in some cycles that fails categorized as a plantation failure or a plantation failure. And the one point which is very important to know in pregnancy, and maybe we all should know this aspect, that the categorization is very simple. That is the chemical and clinical. Chemical means once the acid is detected in the serum or in other fluid of body, that is called chemical pregnancy, and that's the first onset. But doesn't lead to clinical pregnancy, because clinical pregnancy means it has to be viably seen through sonography. So once the heartbeat is attained, that's called clinical significance. Clinical pregnancy in normal settings. So in the egg settings, when this is detected, or the gynecologists find this pregnancy for pregnancy, completion. And in the different factors point of view, we see the ART in the IU, ICSI, they are the first things to be done before we go for IVF. If this fails, then there's a possibility that we have to go too many cycles. And that renders the uterus because of nursery conception and other factors contributing, which is well known, metabolic and endocrine systems problem, obesity, Pcos, or discussed, and also endometriosis, because they give rise the disturbance in the endometrium and makes hostile for those implantation. So if I just take you to the cone which I just pulled out, that the large pregnancy failure happens at the level of uterus. Okay. Yeah, here, fine. So in this aspect, we see that large pregnancy failure leads to see before even they have got the pregnancy setup. And we see not many things development over here, and that's what the things leads to. And this can be because of the factors of endometrium incompatibility, because of implantation failure, chronic endometriosis. Test obviously we linked with all aspects, so I think it's not debatable over here. And also the neurological aspect, because the fetus is having both the parents antigen. So if it is male, then we will have derailment of the pregnancy by way of antibodies. And there are some regimens to treat the recurrent pregnancy or pregnancy failure, where matters is because of anatomical, hemophilia, genetics, all are being taken care. If there's an issue with everything, different factors are taken care. So that means we have something, but still there's some sort of black box, we call it, or maybe say uncertainty developed in the treatment of recurrent implantation failure. We have to look at some aspect which we can bring it to our clinical setup, which will be useful. And people have tried different regimens, and they are still struggling with that thing. And they have tried to go through the therapy, aspirin, different combinations, vitamins, the pentifoxylin, and even also the plasma-rich, administration to the patient who can come and benefit out of it. But this is not still a way of solving the problem. We still have problems persisting. And what are the gaps which we have? That is how the embryo with mother get failed to attach, and how this has to adopt. These things we have to look upon and debate on in subsequent time. And keeping in view of that, there are some clinical findings where they say that if the biopsy of endometrium is taken and if it is analyzed for some sort of polyadipate, I must call it solution, then the PAR comes in picture, and in secondary phase there's a loss. This is normal endometrium, which is the word of PAR, but if it is proliferative, need the polyadipate isolation. That's why the concept cropped up that if there's any role of the polyadipate as a polymer, which is responsible for large polyadipate isolations in the system. And we looked at all these aspects, and one recent publication has come up from the Mayo group that if the PAR1 and PAR2 is knocked off from the endometrium, they can lead to emission failure because of poor implantation as well as restoration process. That way, we drive the hypothesis to see it, and we take in the model of mouse because that is more easy, as we have listened in the morning that we don't have possibility of these biopsies or sample because of pregnancy we don't even allow to have the normal sampling of blood typically. But yeah, with some sort of riders we can have it. And we try to mimic the conditions of implantation failure by way of boosting any specific agent which can delay the implantation failure. So we have seen this knock of pregnancy by of these blue bands of pregnancy, and we find that PARP2 is abolished in these cases. That means there's sort of linkage of PARP2, but PARP2 is not alone to be responsible because PARP2 has to be regulated by substratal mechanism, which is caspase dependent, and we know very well that PARP1, 2, and so on, they have caspase linkage. And the PARP2 is regulated by caspase 8, and we observe that if we see this protein expression of caspase 8, the full length one, we find poor levels, but as soon as the pregnancy is during, we can see this increase levels and also the activity. That means caspase is indeed one of the factors responsible for PARP2 to create vulnerability of PARP1 function in the pregnancy failure or other implantation failure. Then these events would take place, so we are not sure how the PARP2 is playing a role. So we looked all these windows of implantation where we have addition, invasion, so on, so things, and we find that once the onset happens of pregnancy, then the PARP2 start expression, and at the time of implantation where the embryo attached with the mother to have common various physical and biochemical interactions, that leads to increased levels So they said that the PARP2 push itself to nuclear compartment and does function as being classically reported. Subsequently, we analyzed its regulator caspase 8, which is its regulator post-transcriptally. We find that caspase 8, when it is facing the implantation period, is downregulated in terms of its expression and at the same time the activity. You can see over here, this is a typical PARP2 level in pregnancy where we only have the pregnant animal without the embryo. Especially pregnancy where there is no PARP2, but as well as pregnancy where increase over here. And the fact which we have observed, citing that in pregnancy, none of the what you call is milk compartment expresses the PARP2. So we looked at all these model systems and to have whether the PARP2 is indeed required for pregnancy establishment. And then we looked at the regulator, which is caspase 8, as indeed requires for its regulation purpose. Yeah. Yes. So in typical cases where there's So the PARP2 which we studied in different model of pregnancy where we used pseudo pregnancy with a controlled environment of hormones. So that is showing very important role of PARP2 in this condition, and to confirm whether this PARP2 is indeed important for pregnancy, at least in mouse model, because in humans it is still a question because of the limitation of the tissues. We find that once we knock off, as we have Dr. Mali speaking about, the in vitro intraluminal injection of the either siRNA or specific agents, we can abrogate the pregnancy. That means PARP2 is indeed required for given periods. We can see all these embryos viable, but the pregnancy is not set up. And then to confirm whether the effect is because of the embryos, we did before day 3, which is just the time where the embryo start migrating to uterus. And when we administered on day 3, we could not find a disturbance in the embryo. They are all viable, all as normal morphology, but the blood size are abolished. That means the uterine environment required help of PARP2. And to see whether the condition which we have seen in the mouse are happening in the humans. So we adopted a model which is very well known, in vitro reproduction model, where we co-cultured the embryos from and also the human derived cells trophoblast. This is the embryos from mouse, and this is the cell line from human origin of endometrial epithelial cells. We find that once this PARP2 is inhibited by fibrinogen inhibition, we cannot see much growth of this embryo. That means PARP2 definitely is indeed important for embryonisation process in the human as well. And this is just to recapitulate whether the homologous culture can also remove its conditions. Indeed, we find the same thing. And then definitely realize some of the biomarkers and also the factor which is downstream of PARP2, which is confirming the importance of PARP2 in the embryonisation process. And as I said, that caspase-3 is important, so we went ahead and looked at the caspase-8 role in the endometrium. And first, we confirmed that whether caspase-8 can inhibit, maybe classically we know, but whether the signal is operating in the endometrium, we don't know. So we have confirmed. So by using the primary cells and inhibiting caspase-8, we confirmed that PARP8 inhibition doesn't lead to apoptosis, but it does indeed decrease the PARP2 expression. So this is the overall strategy where we have purified these cells confirmed by FACS, and those cells which are positive for Biomentin, they are categorized for triple cells, and those cells which are positive for the cytokeratin, we have categorized for. We find similar findings that caspase-8 indeed, either in stromal cells or cells, they have this function operating. So taking all these things, we confirmed, and then we moved to this post-transcriptional regulation at the epitope levels, and we confirmed that PARP2 does have the 5'-EGR, the binding for amino 49, and we confirmed by nucleotide assay over here. And then we get to see whether the caspase and PARP2 somewhere is linked because of the mRNA expression, and we did find that the mRNA 149 regulates the inflammatory process in vitro conditions. And secondly, we find that when we did FISH, which is the locations of transcript of mRNA, we find that the given period where the inflammation taking place, its levels are decreased. And this is the post-synthesis of fluorescence and so on things. And then, as we know that the EMT has been spoken about, one of the important event for implantation, and just to recall that the epithelials are lost during the implantation process. That's why we looked the ecadrin expression also the ecadrins, but here I'm focusing on ecadrin. And we confirmed that the ecadrin is basically downstream of 149. It is one of the regulators, and we confirmed that when the embryo is somewhere implanted, surroundings are lost because of the ecadrin revolution. So what we see that the amino 49 can control the PARP2 in the luminal cells, stromal cells, which indeed leads to various changes at the PARP2 level, which promotes caspase. We don't know what mechanism, and finally helping in the process of implantation and having the proper dialogue of material nutrient interaction. The team which have been working with the past people, we are the current people over here. We have a lot of fundings. So with the time crunch, I will stop over here, and we'll have to have any questions over here. And thank you all.
To know how this mirror 149 control the PARP expression.
It binds to the UTR of PARP2 mRNA. So by binding, it tries to destabilize the mRNA, so either lead to degradation or repression of the translation.
Wonderful. Thank you.
So we have this because of this some problem of the slides. I could not really focus, but we did find both ways. We confirmed by nucleotide assay.
It's very interesting and excellent talk. I would like to know, by inhibition of this PARP2 leads to increase in number of embryos, but what we should or
if maternal PARP2 is inactivated, it leads to the susceptibility failure of endometrium. Though it doesn't affect the embryos, but there are studies available. Suppose we knock off the PARP2 from embryos, they cannot undergo sperm development. So they die, gestational period, maybe say 10 or so. And if both are knocked down, there will be severe problem. They will have premature gestational failure. It will be absorbed in the uterus.
Are there any studies on the expression of PARP2 in inflammation site in humans or expression of MERU 149 in
somehow because of this my titanation, I could not show. There are studies available that mRNA 149 is expressed in the endometrium and to influence. So people have done some sort of microarray where they have found this mRNA 149 expressed in the human. And the PARP2, as I showed that in the endometrium, it does express that too in proliferative phase. That means when the cells are proliferating, they are expressing. As soon as they attain the secondary phase where the endometrium requires the secondary stage, where we have a lot of those secondary glands formation, so basically is off from the expression level.
Nice talk. Thank you.
Only part are they two are involved in the process, now other proteins are not involved in the complete implantation.
Yeah, it's like a network which we have to rely on because PARP does definitely have a role in the case, but not only directly. Maybe it has some, like I said, that ecadrin is linked with PARP. And caspase 8 is regulating the PARP. So it means there's interwoven connections of molecules or say signatures which probably contours in a one very nice harmony of the PARP2 government.
Can we establish a flow chart that starting and to the end, the signaling and cascades of events happening that is already established, and we can also look into that for implantation, proper implantation, to retain implantation. What are the proteins? So that those kind of things are.
If I am picking your question correctly, the events of implantation are very well established, at least the main events. There are events which are gray, which we know that pregnancy failure. So the embryonic also contributes, and also the maternal tissue. Now those interactions are still, questioned. So there we try to shed some light whether the PARP2 coming from mother origin can be important factor or not. And one article has come up recently in the PNS by D. Mayo, who has shown that, the group has shown that the PARP2 and PARP1, when it is knocked out from the animal, they lead to the resorption of the embryos and also failure of the implantation. And subsequently, because in humans and mouse, the similarity somewhere is there, but there is also dissimilarity where gestation basically happens after the implantation. So those events are very heavily, disturbed. But in this case, we want to have whether genetically the PARP2 regulated and is regulating the catenin, because catenin also is known to have different roles in the time frame of this window. Thank you.
I call upon second speaker, Dr. Suresh Indu, professor from University of Hyderabad, and he is a area specialization in reproductive biology, and he has received ICMR research awards. First.
Thank you, madam. And at the outset, I would like to thank the organizers for giving me this opportunity to present our latest findings. So whatever I am going to present here are very preliminary findings, what we have done in the last 1 year or so. So before I go into the details, I'll just give you a basic idea of what exactly happens during sperm maturation. The spermatozoa which are produced in the testes are immature, and they pass through the epididymis to undergo the process called the sperm maturation. And during this passage to the epididymis, Yeah. So during the process through the epidermis, a large number of proteins are added at the sperm surface, and this makes them to be motile and to acquire fertilization ability. And the epidermal transit takes about 2 to 6 days. And as I said, only those spermatozoa which mature in the epidermis have the capacitation ability. So, and the important thing is that the fate of the spermatozoa depends on the environment in the epidermis. So, it's the microenvironment of the epidermis is crucial for the maturation. And there are a lot of studies which determine the epidermal proteome. They have identified about 250 epidermal proteins, and many of them are added onto the sperm surface, and they also form components of the sperm. They exhibit a lot of different roles, diverse roles, and they are more very important for, as I said, sperm maturation, and some of them are anti-capacity factors. Some of these epidermal proteins contribute to innate immunity, that is by acting as antimicrobial proteins. And interestingly, one of these epidermal proteins, HE4, human epidermis protein 4, is a marker for cancer. Though a lot of information is known about these epidermal proteins, majority of them remain uncharacterized. So we started our work way back in 2006. We identified a gene known as the RAD SPAG11A gene, which is also called as the SPAG11E, which is located on the chromosome 16. This SPAG11E in the rat was later known as SPAG11A, and we have identified, along with this, we also identified two other genes known as SPAG11T and SPAG11C. SPAG stands for sperm associated antigen 11. So this gene is known to be epidermal specific, especially the caput specific. This was reported long back 2001, and they have shown that this gene is epidermal specific. And when it was discovered, it was called as BIL1B. Later it was changed as SPAG11E, and finally it is called as SPAG11A. So SPAG11 is a protein family. There are a lot of isoforms in the sperm associated antigen 11 family, and they have a lot of functions. They are most importantly, these SPAG11 proteins are structurally and functionally similar to beta differences. Beta differences are the antimicrobial proteins, and majority of the proteins of this family are specific to the male reproductive tract. They display antimicrobial activity, and we have also shown that they display synergistic antimicrobial activity with the antibiotics, and they induce endotoxin challenge. They are inducible during endotoxin challenge, both in vitro and in vivo. And the induction is dependent on toll-like receptors, NFKB activation, and androgen receptors. And further, we also showed that induction is epigenetically regulated. As you can see here, this is the difference in 1 and difference in 2. These differences are characterized by 6 cysteine motifs. So similar 6 cysteine motifs are present in these SPAG11 proteins. That's why we consider them as to be defensive-like. So as time passed by, there was a new emerging concept about the role of defenses and cancer. So human beta defensin 1, defensin 2, defensin 3, and HE4, they all have been shown to have some role in human cancers. Human beta defensin 1 is supposed to be a tumor suppressor. It inhibits migration and invasion of cancer cells and downregulation in many cancers, and it's downregulated in many cancers. Similar with defensin 2 and defensin 3, this exhibits anti-cancer properties, and human HE4 is known to be a potential biomarker for many types of cancers. So the proteins which are found to be very specific to male reproductive tract, somehow they have been found to be having a role in cancer also. So and now I change to a different topic, that's between cancer and the defenses. Now there's a very important aspect which has been emerging in recent years. The incidence of cancers in the male reproductive system and the prostate, testes are quite high, but whereas the cancers in the epidermis are very low, it's almost 0.03%, which is quite negligible. So this poses a very important question as to why tumors are rare in the epidermis. You can imagine tumors in almost all parts of the body, but the tumors in the epidermis are very, very rare. So what is that this organ is so special? There have been many theories. Of course, many people have not worked into this area. They say that there is unique properties of the epidermis. It has a number of growth suppressor genes, anti-oxidant production, stable and tight cell junctions, strong immunosurveillance, oncogene expression, and decreased cell proliferation rate. So all these factors are majority of these factors which they propose are found to be evidence specific, and they said and they propose that all these factors contribute to the very low incidence of hepatitis. So we wanted to analyze whether since Spaghetti is similar to defenses and defenses are a role in cancer, so we want to see whether Spaghetti is one of the gene that protects hepatitis from the induction of tumors. So to this test is we did was we initially immunized rats with Spaghetti protein, so that by active immunization we decrease the Spaghetti protein levels in the body. Then we, after once we determine the titer, and if the titer is very good, then we treat the animals with diet and nitrosamine at starting of 16 days and at a concentration of 0.01% in sterile water orally. And this protocol was adopted by standard, the previously published paper. And this water and the animals were subjected to den treatment for 12 weeks. This is a very low dose, and it is known that this very low dose may not cause severe hyperplasia of the tissues. Okay, and the important thing is that the antibody titer was maintained throughout the carcinogen treatment period. So, and once this is done, we have observed the tissues by histopathology analysis. We have observed field analysis, caput, testis, prostate, liver, and the lung. These were then general tissues, liver, and the lung. And I will just show in the form of a table what we observed here. I will focus more on the caput here. This is the control. Of course, we expect normal morphology in the immunized animals. That is where Spaghetti was injected and the antibodies were generated. The tubular morphology was normal. And in the only den treated, it's a very low dose of carcinogen. The caput was found to be normal. Whereas in the case of prostate, testis, and the liver and the lung, you can see moderate degenerate changes when they are treated and the animals were treated with low doses of the carcinogen. So this is a very preliminary evidence to show that at very low doses where the other organs can be affected, hepatitis seems to be resistant toward the tumor incidence. So in animals which were immunized and were treated with the den, it means that these animals, levels of the Spaghetti proteins in these animals are very low. They have been ablated because of active immunization. You can see the incidence of the histopathology report shows that there is occurrence of foci fibrosis, infiltration of inflammatory cells and between the tubules, and severe degenerative changes noticed in mucosal layers of the tubules. So these two observations have been very important, and the very low dose in the normal animals, there is no effect on the caput region, but in the immunized animals, which means that when the Spaghetti levels are very low, then you see there is a development of this. I mean, that's what the histopathology says, incidence of oncogenesis or initiation of oncogenesis. So we thought we'll explore a little bit further what is the molecular aspects behind this changes in the epidemics. So when we were looking as to how to explain this, we stumbled upon a paper which was published in Cell recently in 2018, where they have studied the cancer genome atlas and the common signaling pathways. They have analyzed 9,125 samples, human samples of different 33 cancer types, and they checked all the pathways which are affected in cancers. So they ended up having 10 pathways. These are the major oncogenic pathways, signaling pathways, in which the gene expression gets altered in different types of cancer, most of the cancers. So they are the cell cycle pathway, HIPPO, MYC, NOTCH, NRF2, PI3K, RTK, TGF-beta, TP3, and WNT pathways. So what we thought was, okay, what happens to these pathways, the components of these pathways, that is gene expression of the components of these pathways in animals in the caput region that were obtained from den-treated animals and the control animals. So I am just going to show you 10 tables here because there are 10 pathways. Initially, is the RTK-RAS pathway. So here we are showing you the immunized versus control. In the immunized treated animals, all these genes, they are downregulated, which is shown in red, where the value is below -1. And the same kind of gene expression was also seen in the case of den-treated animals. I hope I'm clear about it. Whatever gene expression pattern that is observed in immunized animals was also seen in the case of den-treated animals. this is an indication. Similarly, there are some genes that are upregulated. Those genes which are upregulated in the immunized animals were also upregulated. Majority of them are also upregulated in the then treated caput. So this suggests that the microenvironment in the immunized animals is similar to that of what happens when tissue is exposed to a carcinogen. So the absence of asparaginase renders the tissue to be more oncogenic, and the expression pattern of genes that are involved in signaling pathways are altered as similar to what is you can see in the case of carcinogenic animals. So those shown in yellow color also show similar expression pattern, but the values are not above below -1 and above 1. Similarly, we have seen in other pathways, the cell cycle pathway. Of course, few genes were, if you look at here, these are the genes which have been upregulated, and some of them are upregulated. And here, this is downregulated here, and similar pattern was seen in many other genes, though the log2 values were not significant. This is the case of BLIC pathway. You can see these genes are downregulated and similar expression pattern, and these genes are upregulated. And the similar kind of expression in the then treated animals. We did not see much changes in the hippo pathway, but we could see some changes in our arch pathway also. Similar changes in both the treatment groups. And here the changes are almost most of the genes have been changed. You see a lot of genes that are downregulated. Similarly, in the case of then treated animals, and even the pattern was similar in both the groups. And the TP3 and the TGF pathways, as I said earlier, number of genes, the gene expression pattern seems to be similar in both the pathways. And this is the WNT pathway. As you see here, a large number of genes have been downregulated, and you also see almost 70% of the genes in the then treated group also downregulated. Same in the case of upregulated. So this yeah, of course, one more pathway, the another pathway. We did not say actually, you can see here the same changes here. So then what we did was let us see what happens in the case of knockout animals. In the knockout animals, for sure, the gene is eliminated and there will be no asparaginase protein. So we generated knockout animals in collaboration with NCBS Bangalore. And the strategy was this is the asparaginase gene, there's 2 exons, and designed this guide RNA such that the exon 1 is removed and a part of the exon is also, intern is also removed, and this is the stag11 deleted version. So in this stag11 animals, we have again done with histopathology analysis, and the report shows that in the caput there is severe mucosal epithelial hyperplasia of tubules, moderate degenerate changes were observed in the mucosal epithelial cells, and cauda seems to be normal between the wild type and the knockout animals. And of course, the testis, there are similar changes which we also observed in the case of stag11 immunized animals. So there seems to be some correlation between the immunized animals as well as the knockout animals. So this is the preliminary data we have generated, and basing on this, we have some conclusions. It is proposed that depletion of this protein in the epidermis alters the microenvironment, which may result in its susceptibility to development of cancer. And factors that regulate a variety of processes to maintain cell homeostasis were reported earlier. And the differential expression pattern of these factors observed in the caput of then treated only animals was similar to the caput of immunized only animals. So this provides additional support to the hypothesis that epidermal specific proteins, stag11 in this case, plays a crucial role in the prevention of cancers in the epidermis. So the statement is off the limit, but I would say this preliminary evidence. And we would also do further analysis by real-time PCR and Western blotting to confirm whatever differential expression we have seen is true or not. Okay. Yeah, I think I'll stop. Thank you so much. And I'll be happy to take any questions from you.
After questions are there, and you are in time.
Thank you so much. Sir.
Appreciate for the informative paper. And I'm wondering, many of the proteins what you are showing are having antimicrobial activity. because of suppression of the antimicrobial activity, that may be leading to the development of cancer. That is one question. The second question is, do the protein have any role in the cell cycle regulation?
This Catecholamines protein. No, till now, I mean, differences are known to have role in cell cycle regulation. They control cell cycle regulation. And this protein is a difference-in-like protein, but we haven't checked the role of cell cycle regulation by this protein.
Any other report from the literature? Do they have any role?
Epinephrine specific proteins, no. As far as I know, there are no proteins that regulate cell cycle regulation. And this protein we have previously shown in vitro also that when you in the primary epidermal epithelial cells, when you downregulate this protein, the cells start proliferating more. So indicating that this protein may control cell proliferation. That was previous work we already published. And then the answer to your first question, since this is a defensive-like protein, and whether that may contribute to the increased incidence of cancers because it's a defensive protein. Generally, some defenses are known to protect the cells from induction of tumorigenesis. So naturally, the decrease in this protein will contribute.
In epidermis, the protein revolving on having something in the epidermis epithelial cells.
This is a secretory protein.
Yeah. So you will have the autocrine and pragmine things.
Yes,
going
both
on.
of them. That's what we think. We are also interested to identify whether there is a receptor for this protein on the cells.
In the sperm.
On the epidermal cells.
Okay.
Because we say that this protein governs cell proliferation in the epidermis. So as you said, it can be acting on a receptor or it can be acting within the cell itself.
Okay.
So that part is not clear.
Okay. So do we have some clarity with this self-life or distance in the epidermal tube?
This protein,
Yeah.
is half-life.
Yeah.
These proteins, when they are secreted, they bind to the sperm.
Okay.
And once they are bound to the sperm and then the sperm are deposited in the female reproductive tract, these proteins are released so that they can act as antimicrobial proteins.
So it's a very interesting and new finding. So I would like to know, are there any protein homologous to these expressed in normal cells?
I can't get it, sorry.
So similar type of proteins, homologous protein. Are there any homolog of these proteins expressed in?
Stag11 proteins. Yes, there is Stag11 B. There's other proteins.
That are expressed in normal tissues. No.
This protein is epidermal specific, but most of the Stag11 proteins are epidermal specific. They are not expressed in other tissues.
Okay.
But there are few proteins which are expressed in other tissues also.
Okay. Are there any studies where treating the cells, cancer cells with this Stag11 protein, and they have found any anti-cancerous effect? Or are there any studies?
So what we did in our earlier study was we have taken epidermal cell line, and it's not a true cell line, it's a motor cell line. We have overexpressed this protein, and the cell proliferation decreases. So it clearly indicates that the presence of this protein itself decreases cell proliferation. But with the differences, yes, they have done adding differences to cancer cells and shows decrease in cell proliferation. But we have not done such experiments.
Interesting.
I think we can stop here. Other questions can be discussed in person and for a different
Sure.
talk. May I call upon third speaker, Dr. Chaturaj. He will be delivering a talk on the incidence of light on the feeding behavior of zebrafish involving gut and brain. He is a professor from Department of Animal Sciences, Kazi Nazrul University. His area of specialization include chronobiology, and he has received MRE award. So may I request Asmanda to give a talk?
Thank you, ma'am. At the outset, I should thank to the organizer, and SLBC for all of us is just like a second home. And post-lunch presentation is always good for the presenter because so anyway, let us come to the business. So from the starting of today's talk, Professor A.V. Ramachandran sir was talking about the meditation and synchronization. Obviously, that is the main clue. Whatever we are seeing here, whatever the pathways we were talking about, it should come to a particular signal in a particular time. Otherwise, biology is nothing but a particular signal in a particular time. So if you see in these two pictures, you can clearly see the differences. One is in the normal daytime, and this picture is not where the daylight is almost gone, but the night has not started. So in between that, and you can see that there are some lights, and obviously these two pictures, if you closely see, it gives a different kind of sensation to us. So that artificial light is coming to the place. So if we go back to the, as I was talking about that a particular signal in a particular time, and we know that all organisms are having the signals, and right action at the right time is very important. And for this particular thing, we know that circadian system or any kind of biological rhythmicity, which is very, very important for any kind of organism. Either it is an unicellular organism or it is a multicellular organism. Because as we know, the introduction for there is no definition of life, but life is having some property. And one of the most important properties is the response. And this responsiveness, either it is a unicellular organism or a multicellular organism, it is always sustainability and the development of that particular organism, how it is interacting to its environment. So, and here lies the study of the chronobiology, the timekeeping mechanism. And if I go back to the origin of light, as we know, the 24-hour light and dark cycle, which is only prevalent in the earth, and this is the one of the major reason to origin of life in this earth. So, and as I was talking about the particular signal in a particular time, and where clock system is very much important, and if you see in the cyanobacterial also, there are clocks, and which is in the form of phosphorylation and dephosphorylation. And ultimately, when we evolve, the clock system become very much complicated and very much synchronized too. So these are the major clock, which we know that the clock is the circadian locomotor output kaput. We have the BMAL, brain and muscle unity. We have period. We have cry, and this particular system, if we see that this clock system, as we have told, okay, it is regulating many functions. And as we are talking about the chronobiology and the chronobiotic molecule, we know all that melatonin, which is one of the students was presenting in the last session about the melatonin, and which is a chronobiotic molecule, and we all know that it is only synthesizes more during the dark, and irrespective of the diurnality or diurnal or nocturnal, melatonin is always high during the daytime and during the nighttime. And the reason behind is that there is an activation of the N-acetyltransferase enzyme, which is light sensitive. It only actually activated when there is darkness. So that's why melatonin is called as a signal of darkness, or the chronobiotic. Till date, the only chronobiotic molecule we are having, which is melatonin. So now, if I come back to the clock mechanism, how this clock mechanism, and this is our study, and also very few people in other areas, we have shown one thing, that this melatonin and this clock system is mainly coming with the transcription-translation feedback loop mechanism. So till now, there are several clock genes are available, but this clock, BML, PAR, and CRY, these four genes are making this transcription-translational loop. And you can see that when this BML and clock, they are coming into the secretion, they form a dimer, and they activate the another CRY and PAR. And when this positive CRY and PAR is forming, it is giving a negative signal to the BML and clock. And ultimately, the secretion of BML and clock goes on. So this is the feedback mechanism. Once you have the BML and clock activated, it is activating the PAR and CRY, and this PAR and CRY ultimately going back to inhibit the BML and clock. And other proteins, like if you see that ROR alpha and REV alpha, there are other two factors which also influence the BMN clock. So this is a completely again a synchronization process where BMN clock and PAR and TRI are playing a major role over there. So now if we go, so as Professor Ramachandran sir was telling in the morning about the meditation and the role of the nervous system. So basically this, in case of multicellular animal, initially it was thought that this all synchronization process is coming to a master mechanism. That brain is the master, and brain is telling every peripheral organ of a multicellular organism that you do this and you don't do this. But nowadays, we have proved that no, it is not like that. All the central and all the peripheral organisms of a multicellular organism is having a clock. Each and every cell of your system is having a clock, and it is cross-talking to the central system. And this is a particular thing, and we have also shown that melatonin, the synthesis of biosynthesis of melatonin, is a key regulation for this clock synchronization. So, and now, if we come to the artificial light, from this 2013, we have the data, and if you see what is the condition of artificial light at night in respect to India, and this is really alarming. And every day it is increasing because we think development means light, and we are forgetting that natural darkness is the prerequisite of life system. We are forgetting. So, and the most important or most alarming thing is the identification of the LEDs, because we know LED, it is basically a blue. So 450 nanometer, which is the range, I mean that the wavelength is very small and it has a very, very high penetration power. So it is really, giving a lot of clue to our, but till date, we are not able to, categorize light as a pollutant. But in four countries, we have submitted our plea to their parliament to, give the light as a pollutant. So in our early study in last year in Baroda, I have presented this work about the desynchronization of the peripheral ovarian system, that how the ovarian system is being totally abolished, the system is totally, and how this artificial light at night, which is called the ALAN, is destroying the ovarian system. So now I'm coming to the mechanism of the feeding activity. We all know that there are some agents who are responsible for the feeding and non-feeding. So when feeding is important, really we know one of the major important molecules which is important for the feeding activity, and also the leptin is another molecule which is against the living. But there are other, so which is promoting the feeding we call is orexigenic molecule, and who are inhibiting the feeding we call as an anoxygenic molecule. So how this orexigenic and anoxygenic molecule in the influence of light, how it is being influenced in our system. And we also know that this feeding fasting cycle is very much important for the energy balance and our homeostasis, and it is all known. So if you are in a very good system of the feeding fasting cycle, so your energy homeostasis is maintained. And now we want to see that how the brain and gut, because this feeding mechanism, what is happening inside our body, it is not only the function of gut only, because brain and gut both are cross-talking each other. As I was talking, that it is a total orchestrated phenomenon. So brain and gut both are involved in this particular action of maintaining the homeostasis. So what we have done, we have taken the animal, because this work is mainly on the zebrafish. So we have done, feeding was not interrupted. 3 times we have given the feeding, and then after 24 hours of normal light and dark, we expose the animal, either it is continuous light or continuous darkness. And then what we have found in brain, if you see that this is the normal energy cycle, and this is the clock, this is the BML, this is the clock, this is the TRI, and this is the PAR. So you just observe the pattern of cyclicity over here, normal like schedule, and if you change it, you see suddenly there is a change in the expression and also in the peak time. So coordination is lost. Here you can see directly when you are giving continuous light, this coordination is lost. And when you are giving the dark, or even the coordination is lost, but that is not that much lost. But light, it is really, and if you see the another two molecules, leptin and M4, which is an important precursor for the feeding activity. If you see when there is normal light, this is the situation, and when you are giving, you see what is happening when you are giving the light. So total desynchronization of the breathing. Now, if you see in the dark also, there is a problem. But if you see the continuous light, light is influencing more to the effect. So, and same thing we are finding when we are observing in the brain, we are starting that yes, leptin is happening the same way, the expression we have seen in the mRNA, we are seeing the same thing over there. And also the code, which is a acetyltransferase, because it activates the valine. So we are seeing the same thing happening over there. So now, and this is the, so you can skip this, and this is, and also when we observe the gut somatic index, that the content of gut and melatonin, we are finding that okay, when there is a fooding time is more, at that time melatonin is. So that means it is mainly depending on the food mechanism. But here clock is ruling the mechanism of feeding. So so this is the thing we have found in brain, and that this kind of synchronization of these particular molecules of orexigenic and anorexigenic is lost, and ultimately what is happening, the feeding is more in case of light, because as the leptin and other molecules are less, which is controlling the negative, because as I was telling that negative regulation is the most important regulation in case of any biological function. When your leptin is being inhibited, so the whole mechanism of feeding activity is increased, and that's why we are getting overweight animal within a very few days of time of exposure. So when an animal is in the light condition, it is taking more food and it is becoming obese. So now we are going to see about the melatonin biosynthesizing enzyme in case of gut, because it was not known properly that at which position of the gut these melatonin biosynthesizing enzymes are present, and we have identified the main two enzymes, the N-acetyltransferase and that the ASMT or hydroxyl-methyltransferase. So these are the present, and when we are seeing that melatonin is present over there, and you can see when in the normal GSI is increased, and at the time of taking of food, melatonin is more. So melatonin is very much working along with the feeding activity. So there is no problem with the melatonin. But when we are again seeing what is happening in the gut about those clocks which we have seen in the brain, we are seeing the same thing. This is the normal light and dark condition, and when we are giving, you can see here this power one, and also this cry, and also this, you can see there is a shifting in the rhythm. So this transcription translation feedback loop, which is the main key for any cell's organization and any cell's reaction, that is being lost over here again in the gut. So we have seen in the brain it is lost, and again in the gut it is lost. And similarly, if you find the leptin and NUCB2 or the nesfatin, so these are the against the feeding, so it inhibits the feeding, and you can see this is in the normal light and dark, and when the same thing in the valine and M4, which is the activator. So here in the both case, there is an increase in the valine and there is a decrease in the leptin. So, and the mismatch in their regulation of timing, where they are falling, because the peak is very much necessary. When there is the peak is not in the proper time, then the feeding will not be done. So this also shows that the same thing, and this is the hypocretin, which is a foraging, responsible for the foraging behavior. And we are also seeing when there is light, this foraging behavior is also changed. That means is inhibited. That means when it is getting more food, it is stable in a particular area. So this is what we are if you see that melatonin clock gene, and these are very much, sorry. So when light is there, you can see the feeding behavior is increased, and when there is dark, it is less. But when you compare in between light and dark cycle, this particular clock associated gene, along with the synthesis of melatonin, which is obviously low in case of light exposure, it is being hampered. So, and ultimately, if you see the artificial night at night, what is actually is dying, it is hampering all the system in an environment, and ultimately the functionality and stability of the system is lost. Because if your clock system is not properly working, then every system of your ecology will be hampered. So this is a very and if you see a healthy clock and a disrupted clock, what kind of parameters, and now we tell it, very proudly that okay, we are suffering from the lifestyle disease, but lifestyle disease is nothing but the desynchronization of your clock, because a particular signal in a particular time is not being happening. So that is being proved by our study also. So this is my lab mate. So these all people are worked over there, because and they are now in various areas of their study, but I'm very sad that one of my PhD students who was supposed to submit her thesis in November, she died in October 19, and in this communication, her contribution was more, but we lost her. Thank you.
Discussion or not for question?
Yes, thank you, sir, for the talk. Sir, I just wanted to know what is the status of, have you checked about regarding the AGRT and POMC status?
No.
Okay.
Because as we have checked the ghrelin, piperoghrelin, both, all the pathway we have checked. We have not checked insulin also, but these are things why we have checked, because these are the pathway which people, because our next aim is to see that, because when we are seeing something about the gut, so gut is they have lot of factors are influencing, and when you are talking about melatonin and clock, obviously you need to show that gut microbiota, right? So that is our studies going. We are going to communicate that thing. We have got a very important clue on this. So we have not checked. What you are taking, we have not checked.
When we talk about the clock genes, they have now been shown different nervous systems. Talk about brain, talk about we talk about individual. Now, if I just take you back to your slides and want to understand whether these genes are going to be specifically present in the hypothalamus, or they will have somewhere going to other systems.
In each and every cell of your tissue, each and every cell it is present.
Right. So whether the light is triggering the action through melatonin in the brain, or will also affect the other system parallelly?
It is melatonin and clock. So melatonin and clock, these are working synergistically, right? So if you see the other work, when the melatonin pathway is on, then it is showing that the clock activation is starting. But this relationship is not equal in every time. Because that has also an important factor. If it is equal in suppose when you are hungry, at that time it is showing that these two relate equally. But during reproductive periodicity, they have some mismatch. That is also regulated by other hormonal activities. But it is obvious, if your clock is being disrupted, your melatonin is also being disrupted. That is 100% proof. Not only for as it is for the, I mean, not only for the lower group of animal, it is for the higher group of animals also.
Thank you, Dr. Catecholamines, for the nice piece of work and presentation. On behalf of the organizers, SRBC, I thank all the invited speakers who have delivered this.
Thank you.
Now let us move on to the oral presentation by the scholars. And on behalf of the organizing committee, I would like to make an announcement. Of course, it's a reminder. All the student presenters are requested to become member of the Society for Biology and Comparative Endocrinology, without which your presentations will not be considered for the award of prize. Right? Please make sure that you are member in the society. Thank you. And you are given only 10 minutes for presentation. Right? Please stick to that. Thank you.
Good afternoon. Guys, we're talking about puberty onset in legs and possible expected melatonin interplay.
This way. Yeah.
So now talking about puberty, puberty is considered as one of the major life events in the body. Right? It is marked by maturation of sexual organs. There occurs a lot of hormonal changes throughout this period. So these hormonal changes are brought upon by the HPG axis. So now talking about, we all know about the HPG axis, the pulsatile release of GnRH, which further stimulates the release of estrogen and LH, and further bringing the maturation of the sex organ. Now talking about Cisplatin. So Cisplatin is majorly expressed in the alpha and the LPG region. So the Cisplatin neuron further stimulates the secretion of GnRH, which is brought upon through GPR54. Now the literature about Cisplatin is that it was first found through individual studies when they found that mutation of Cis1R gene leads to impaired onset of puberty and hypogonadotrophic hypogonadism in humans. It was also found the stimulatory role of Cisplatin by expression of GnRH by Ronigel and Kelly. Watts et al. And Abrosky et al. Showed that the majority of expression of Cisplatin is predominant in the arcuate nucleus. Now, talking about melatonin, the previous presentation talked a lot about melatonin and its importance. So the level of melatonin increases significantly at the night time, and they also decreases during the light period. So the study conducted by Selman and group has shown that melatonin level decreases significantly as the adolescent progress. So in a study, also Lima et al. Showed that exogenous melatonin showed antigonadotropic. Now, as shown by two scientists that in children with late puberty, high number of melatonin secretion has been reported, and similarly, low levels of melatonin was observed in children with precocious puberty. So, with around all this data we have, we found that there is a significant gap in understanding how melatonin affects puberty and its role if it affects the reproductive system. So, according to this, my hypothesis states that the level of melatonin before puberty is significantly higher compared to the onset of puberty. And when the onset of puberty occurs, there occurs a decrease in the level of melatonin, which further stimulates the Cisplatin and the GnRH. The objective of this study is to understand the role of Cisplatin melatonin in female scarlet exposed to different photoperiodics. Now, my methodology of the study is weaning rates of 21 days were taken, female scarlet, which was group 6 per group. The treatment was as described. After 21 days, we found the rates were sacrificed and further samples were collected. Sample for blood collection was there. We dissected brain for RNA isolation and further real-time PCR and Western blot was there. We had taken ovaries for the histological study. The results, we found that there was significant increase in the body weight in the long leg group compared to the control. We also found that the gonadosomatic index of the long day group. So we found that in the long day group, the gonadosomatic index we found it to be higher compared to the control group. Now, so the changes in the hormonal levels, we found that in the long day group, the levels of FSH and LH were significantly higher compared to the control, whereas there was a decrease in the short day group compared to the control. The levels of melatonin were significantly higher in the short day group and compared to the control, and the long day group had a significant decrease compared to the control. The data suggests that the long day exposure led to the increase in the titer of FSH and LH, while decreased titer of melatonin was obtained. The gene expression study showed that the melatonin receptor in the short day group was significantly increased compared to the control group. Whereas the KIS1R, that is Kisspeptin receptor and GnRH, it showed in the long day that there was a significant increase compared to the control. We also noticed that there was a non-significant decrease in the short day group in both KIS1R and GnRH. So now, further talking about the protein expression, we found that in the melatonin receptor in the short day, we found a significant increase compared to the control group, whereas there was a significant decrease in the long day group compared to the control. Talking about the Kisspeptin receptor and GnRH, we found that the long day group had significant increase compared to the control, whereas in the short day group we found significant decrease compared to the control. Our histological study showed that there was a significant increase in the number of follicles, which we observed at 20 magnification, whereas in the short day there was a very low number in the number of developing follicles. So, for the discussion, our study shows that different photoperiod has effect on the pubertal onset in female life, and this effect occurs in the interplay of Kisspeptin and melatonin. It was also reported that in short day photoperiod exposure, there occurred an extended period of melatonin secretion, which was reported by level 58 hours. Our results demonstrate a significant decrease in the circulatory levels of melatonin in the long day photoperiod regime, while increase in the short day photoperiod, which is shown by mouse and neutral, but this was reported in the hamster. So in long day photoperiod, there was increase of KIS1R expression, whereas decrease in the short day photoperiod. Similar studies were carried out Bolan et al. And has reported decreased expression of KIS1R in the short day. So our histological study showed that number of follicles in the long day group were higher, in the short day group photoperiod it was lower. To summarize my study, it shows that there was increased expression in the long day group of Kisspeptin, GnRH and FSH levels was higher, and also there was a higher number of follicles. And in the darker phase, that is the short day group, the melatonin levels were found to be high. From the findings of from the findings of this study, I conclude that different photoperiod exposure had an effect on the pubertal onset. We have further showed the role of melatonin and Kisspeptin on the pubertal onset. To further validate the preliminary results that we found, various downstream signaling molecules will be checked, and further immunolocalization for the candidate proteins will be performed. So, for acknowledgement, I would like to thank Norepinephrine University for providing the infrastructure, DSC sub for the project fellowship, and Dr. Vikram Sara Institute for helping me carry out the gene expression study. I also like to thank the organizers for giving me the platform to present.
Now the library is open for discussion.
Clarification. In the abstract, you have talked about FSH receptor and LH receptor. So I didn't find anything here. LH receptor and FSH receptors in the hypothalamus you have mentioned in the abstract. So there is no such data presented here. You're not able to complete it in time, or it's by mistake?
No, sir. To limit it within this period, there was further also a lot of data which is not in the slide.
So hypothalamus is having LH receptors and FSH.
So, we check the hormonal levels for the LH and FSH from the blood. Only the hormonal levels were done.
in nutshell, if I ask you, what is the novelty or what is the outcome of your work? Please, what is it?
What this study is specifically saw on the pubertal onset, not the pre-pubertal or after the pubertal onset. We were trying to check the role individually, histamine and melatonin, and when the study further progresses, we would check the both histamine and melatonin interplay together. And similarly, this work was mostly done on fish and the hamster, and very less data is there on rats.
No. I think we will see. It is not like that.
Okay, Shah, you can have a one-to-one discussion after this session, right? Thank you for the nice presentation. And let us move on to the other oral presentation 5 by Nitin KR. The topic is influence of chronic psychological distress on hormonal interplay in causing PCOS.
Something here.
So there were last minute cancellations. So that's why.
Whether his abstract is available in
Yes, it is there. There were last minute.
number. What is number?
16.
You say that. There's a change.
Yes, sir, because people cancel.
That's fine. If there's a change, please inform in advance, sir.
We couldn't update in the abstract because it was already published. They canceled last minute.
You should have told me that what is it? Okay, 16.
Good afternoon. I am Nitin Kya from Division of Molecular Biology, School of Life Sciences, JHS Academy of Aerospace and Defense. Mysore. My topic is influence of chronic psychological distress on hormonal interplay in causing PCOS. First, what is stress? Stress is body's non-specific response to any external demand where the response may be physical or psychological. There are two categories in stress. One is physical stress, another one is psychological stress. Here in these two categories, psychological stress has been mainly focused. It includes depression, anxiety, fear, and so on. So these psychological stress may be because of familial or work or targets and deadlines, health, and from the society. Why is this so important is that its ability of causing the health issues from coronary heart disorders to metabolic disorders, including reproductive dysfunctions also. One of the major reproductive dysfunctions in women that we are failing to address from decades is the polycystic ovarian syndrome or polycystic ovarian syndrome. Polycystic ovarian syndrome is one of the major endocrine disorder associated with the ovarian dysfunction. The disease is associated with the symptoms like hyperandrogenism, hyperinsulinemia, high risk of type 2 diabetes, then elevated levels of LH, decreased level of FSH, and irregular menstrual cycle, etc. Current knowledge on the disease have ruled out the direct relationships of genes, obesity, and dietary nature of the patient in response to disease prognosis. Several studies have several studies have shown the correlation of PCOS with the psychological distress condition of the patients, which give a data that around 91.9% of the PCOS patients are under psychological distress. A study has given a data that highlights greater than 8 score in general health questionnaire 28 test for psychological stress in the patient, which represents the high anxiety level in the patients. This has opened up a debate emphasizing socio-psychological stress as one of the major factors to be considered in research. PCOS is an hormonal interplay of glucocorticoids and catecholamines, wherein cortisol, a primary stress hormone, under chronic elevation is known to lead to conditions such as hyperandrogenism, onset of obesity, hyperinsulinemia, cystic ovaries, delayed menstrual cycle, interruption in follicular development, and so on, which is very much common in PCOS. Next, this is the diagrammatical representation of chronic stress can lead to the ovarian dysfunction as well as PCOS. The flow is explained in further slides. So next is here I have some tables which explains the role of stress hormones in GnRH, LH, and insulin regulation along the reproductive axis. We are marking the table.
Table.
He doesn't have a table.
Next one. Table explaining the roll up.
Yeah.
He doesn't have a table.
It's not there.
It's not the table is not there.
It's not there. Title is there. Click. Just click. Just.
Okay. The first hormonal family is the glucocorticoid. In glucocorticoid, the first hormone is cortisol. Cortisol can lead to the in direct relationship. Cortisol can suppress the GnRH as well as LH pulse, and it also interrupt the gametogenesis. And in other role, cortisol can lead to the hyperinsulinemia and insulin resistance. So which will play a major role in reproductive axis. So next hormonal family is catecholamines. In catecholamines, first one is dopamine. Under low concentration, dopamine elevates the LH as well as lead to the increase in insulin expression. Under high concentration, dopamine decreases the LH pulse as well as decreases the insulin production. So the main observation we have to do is under psychological stress, dopamine's level will be decreased. So next hormone is epinephrine. Central epinephrine has a direct role of inducing LH, or it upregulates the LH production, and it blocks the insulin secretion by blocking alpha adrenergic receptors. Next hormone is norepinephrine. So norepinephrine has dose-dependent manner in stimulating the GnRH. Means if the higher concentration of norepinephrine is there, then GnRH expression will be increased. Elevated norepinephrine will block the insulin by inhibiting adenylyl cyclase activity. Next is the growth hormone. There is no direct effect of growth hormone on GnRH, but elevated growth hormone can decrease the LH pulse. But in the case of chronic stress, growth hormone level will be decreased, so LH will be increased. So it acts on phosphatidylinositol kinase 3 and thereby inhibiting insulin. Next hormone is the prolactin. Elevation in the prolactin level will decrease the GnRH pulse and increases the insulin secretion at the pathophysiological elevation of prolactin. So this is the overall picture what I have explained in previous slides. So chronic elevation or chronic stress can elevate the glucocorticoids, catecholamines, and dysregulates prolactin as well as growth hormone levels. So in circulation, increased insulin concentration can upregulate the GnRH pulse and can cause the polycystic ovarian syndrome or ovarian dysfunction, and other hormones in the circulation, dysregulation in the circulation either suppress the GnRH and LH activity, or it may increase the GnRH and LH activity, so that it can cause ovarian dysfunction. So current research and future perspective of the study is, most recent studies have highlighted the involvement of the psychological stress in the prognosis of the disease. Although the studies have given the picture about the role of psychological stress in association with PCOS, the molecular underpinning of these two are not yet clear. In future, studies are required to reveal the role of stress-induced hormonal dysregulation in causing PCOS by means of epigenetic regulations that these could cause. Thank you.
Now the paper is open for discussion.
Your finding, what was your study?
My study is induction of PCOS to the rat models by means
You
of.
have not to mention anything about that.
Where is the data?
Sir, actually, I'm started my study just now.
It's a review.
Yes,
Review of
sir.
literature.
It's my review. Based on this background, I have started my study.
Okay.
Okay.
All the best.
Thank you for the nice presentation. And let us move on to the third and final paper of this particular session, OP6. Deciphering the dysregulated genes and pathways of liver and ovary in high-fat diet-induced obese female rats by Kaushiki S. Prabhudesai.
Good afternoon. I'm Kaushiki Prabhudesai. I'm thankful for giving me opportunity to discuss my work that has been carried out at Biomedical Informatics Center at ICMR National Institute for Research in Reproductive and Child Health. So the title of the work that will be presented today is Deciphering the Dysregulated Genes and Pathways of Liver and Ovary in Higher Fat Diet-Induced Obese Female Rats. So basically, in this project, we are attempting to understand the effect of differential regulated genes and pathways on the overall health and functioning of liver and ovary under obese condition. So just how it is happening? I'm not doing anything. I'm going
No, that's why I'm monitoring because
You
they're
are
not.
doing?
Yeah.
So before going ahead, I would just like to give brief introduction about obesity. So according to WHO, the overweight and obesity are defined as abnormal or excessive fat accumulation that presents a risk to health. So as we know, obesity has been associated with several comorbidities, and that has adverse impact on various vital and reproductive organs in the individual. To name few, like coronary heart disease, non-alcoholic fatty liver disease, type 2 diabetes, the development of several types of cancer, as well as the development of polycystic ovary syndrome in case of obese female that leads to subfertility. So according to 2016 data, globally 15.1% females and 11.1% males of age 18 and above were obese, indicating that the prevalence of obesity is more in case of females as compared to males. The next graph is the biology, the geographic distribution of overweight and obesity among the women in different states of India. This is 2015-16 data. So if we see overall this, if we collect the data, we understand that though the prevalence of obesity is more in case of females, the association of obesity with different comorbidities has been well established in males as compared to, and very little is known about females. As well as, though we know that obesity is associated with different types of comorbidities, the crosstalk or cross networking between these comorbidities is not yet well understood. So the aim of this study is to evaluate the effect of obesity on regulation of disease-associated genes and pathways in liver and ovary of female rats. So the objectives set to achieve this aim are development and validation of high-fat diet-induced obese female rat models, and study the impact of obesity on genes and pathways associated with liver and ovarian disorders. So the methodology includes 21 days on immature female visceral rats. The rats were randomly divided into two groups, lean and obese, and then they were fed accordingly for 17 weeks. The rats were sacrificed and blood was collected to perform different assays. Coming to the result section. So first, we wanted to see whether the obesity has been established or not. So we monitored several parameters, and as we could see, the final weight of the animals in the lean group, that is at the end of 17 weeks, the animals which were in the lean group had less, I would say, the final weight of the animals in the obese group, that is at the end of the 17 weeks, was significantly higher as compared to the weight of the lean group, indicating that high-fat diet could induce obesity in these rats. As well as we checked for the adiposity index, and we could see the adiposity index was higher in case of obese rats. Next, as expected, the fat deposition was observed to be more in case of obese rat as compared to the lean rats. Next, when we sacrifice the animals and monitor for the weight of all vital organs and the reproductive organs, we found that in case of obese animals, the weight of the organs was reduced as compared to the lean group. So we saw the establishment of obesity in case of this female rat, and we checked the other parameters including several hormones and the lipid profiling, and we could see that in case of obesity, the serum insulin levels were high in case of obese group as well as the glucose levels, indicating that obesity could induce hyperinsulinemia. As well as when we check for the lipid profile, we could see there were significantly higher levels of triglycerides and serum LDL and lower levels of HDL, indicating that there was development of dyslipidemia in these animals. Then we wanted to see what is the effect of obesity in the genes, that is into two distinct organs. One is the liver and one is the ovary. Liver is the vital organ as well as it's actively involved in the metabolic process, and one is the ovary, which is a reproductive organ. So we perform microarray analysis with these two tissues, and we got there are certain differentially expressed genes in these two organs. Then we check for common dysregulated gene, and surprisingly, we found that there are 125 genes which are commonly dysregulated in ovary and liver. Out of this 125, 80 were upregulated, 18 were downregulated, and 27 were differentially regulated. What do I mean by differentially regulated is, out of this 27, 24 were upregulated in ovary and downregulated in liver, whereas 3 were downregulated in ovary and upregulated in liver. These are further shortlisted for validation using qRT-PCR and nanostring method. So the genes that are commonly dysregulated in two distinct organs is liver and ovary function as the predictive key indicators of hepatic and ovarian health in obese females. Then we wanted to see what is the effect on the pathway, if there is any common pathway which is getting affected in these two distinct organs. So again, we had the data for the DGs. These were mapped with the analyzed for the pathway enrichment using K. And we found that there is one common pathway which is getting dysregulated, and that is retinol metabolism pathway. Further, even the genes involved in retinol metabolism pathway are to be validated. Now, till now I talked about the common genes. Now, further, we wanted to see which are even the candidate markers for the disorders which are specific to the organs, like ovary and liver. So, we actually analyze the data for these two organs independently also. So as I mentioned before, so 125 genes we found to be commonly dysregulated in ovary and liver. The 4,325 genes were only dysregulated in the ovary. We took all the genes and then we mapped the genes to the diseases which are specific to the organs. So in case of ovarian disorder. And we found that there were so many diseases which were associated with these genes. We filtered out only those which are related to the ovary, and we got the information about this. Because of time concern, I will just move to the first two, is hypogonadism and polycystic ovary syndrome. So this is the regulation status of the genes which are associated with hypogonadism and PCOS. So this is the green the genes which are in green are down regulated, and the ones which are in red are up regulated in case of ovaries of female rats with obesity. Further, we have few markers of PCOS. So this is actually ongoing process. I could finish with the two primers of these PCOS markers, and with few RT-PCR I found that the AMH and PNFS levels were high in case of ovaries of obese female rats. Similarly, we performed the same analysis with liver. In case of liver, again the 125 were common in liver and ovary. Remaining 327 were also taken care of, and then we again mapped these genes with the disease specific to the liver. Also, we filtered for the liver disorders, and we got the disorders which are associated with the liver. So this is about the fatty liver disease, and this is about the liver fibrosis. So this is the regulation status of the genes. That this is the, as I mentioned earlier, the down regulated and one in the red are up regulated genes which are involved in this particular diseases, and these are getting up or down regulated in the ovary the liver of the obese female. So the genes dysregulated in hepatic and tissues could play a significant role as early onset marker for development of cluster metabolic disorders in obesity. We are currently expanding this work with second batch. And for first batch, due to lockdown, we could not perform many studies, like we could not monitor the cyclicity, and hence we could not estimate serum levels of gonadotropins and steroid hormones, which are highly dependent on cyclicity. We are performing this in the second batch, as well as the first batch we only saw the effect on the liver and ovaries, but now we are collecting all the vital organs and see what is the effect of dysregulated genes and pathways in other vital organs. So after this, we propose that study would help to identify the mechanistic insight on obesity-related comorbidities. This would help to identify novel biomarkers for early diagnosis of cluster of metabolic disorders under obesity. And further, these novel biomarkers could be developed as pharmacological drug targets for therapeutic purposes. I'm thankful to the Department of Health Research and ICMR, Indian Council of Medical Research for funding. I'm thankful for the staff who are actively involved in this. Samiksha, Tanmay, Sanket, Dr. Vikas, our collaborator, and Dr. Susan Thomas, the PI of this project. Thank you.
The paper is open for discussion.
Yeah, I wanted to know, was there any specific reason for you to choose a female model of that?
Yeah, so when we, as I said, the prevalence is more in of obesity is more in females, but the data is moreover concentrated on the males.
I am referring to the animal model.
Yeah.
Is this an established model or you have established it by yourself?
No, this is the established, this is actually, the research is available, the research article is available. We have used the same
Which
feed.
strain of rat is it?
This is Vistar female rats.
And what diet have you given it, fed the diet to make it obese?
So this is 22% fat diet, and in case of lean, it was 6% fat diet. This was actually selected from the already published data.
And what was the inclusion exclusion criteria for establishing obesity?
So
because the thing is, when it comes to a study which is as long as this, it can very easily revert back, and you can get a false positive or a false negative.
Correct. So we monitor.
And second thing was that we were more interested in seeing the data rather than you just show the number of genes that have upregulation and downregulation.
So sir, that is going on. Actually, this is the this is in the first phase of study. We could finish till now only this, the micro analysis. But as I mentioned, the genes which we have found as well as the pathway which are validating using PCR as well as some other validation techniques like nanostring. So that is still there. So I could finish till this for.
Yeah, but you have told that your blood parameters, FSH and LH, got down because of the lockdown. But simplicity, if you see the logical way of establishing anything, if you have done those things early, then we go to the genetic level. No?
Yeah.
And
so
whether your animal is obese or not, whether other parameters are influencing or not, without showing that you are directly going to genes. So I think that is something.
No, so we have checked the, we have actually.
I'm sorry to interrupt.
Yeah.
We can have a discussion in detail.
Okay.
Induced type 2 diabetes. It is nothing but hyperinsulinemic insulin resistance, right? But in your case, the data on insulin shows significant increase.
Yeah. Correct.
But no change in blood glucose.
Glucose is slightly higher. It's not significant.
It's not significant.
It's not significant.
Right?
Yes.
Please check.
Yeah. It's not coming
If
significant.
it is normal, right, it should be better. That increase in insulin cannot be tolerated by your rat. That would lead to hypoglycemia, and it is better. So please check the data on blood glucose.
Okay.
Right?
Yeah.
Okay. Thank you for the nice presentation.
Yeah.
Good luck. So, in this particular session, we had 3 invited talks and 3 oral presentations. All the presenters did well. So I congratulate all the presenters for nice presentation. More than that, I would like to appreciate the participants for active interaction with the presenters. Thank you. And I get this opportunity to thank the organizing committee, myself and the co-chair for having given us the opportunity to chair the session. Thank you.
Thank you, everyone. I'd like to request the chairs to award the certificates to the presenters. Dr. Asamanja Chaturaj, Mr. Hashisha, Mr. Nitin KR, Ms. Kaushiki S. Prabhudesai, and as a token of gratitude to the chairs, I'd like to request Professor Shivananda Pasar to give the mementos to the chairs. all of you that the dinner has been arranged in Vice Chancellor's residence for the evening. So we'll be leaving from here at exact 7:30. So please gather here at exact 7:30. The transportation has been arranged and the vehicles will leave at 7:30. So now we'll break for tea. Also, Gautam C is requested to submit his PowerPoint presentation. Gautam C. The session 3 will begin at 4:15.